Okay, so here's my screen, and here's the exam. Can everybody see the exam here?
Yeah.
Yep.
Okay.
Yep.
Question 1. Which of the following genes is not activated by p53? So Puma is, we know. MDM2 is, P21 is, right? Because MDM2 and P53 are reciprocally activating each other. ATM is upstream of MDM2, so that is not regulated by P53. Or it is not. I guess it could be, but it's not. Which type of microtubule of the mitotic spindle uses dynein? So dynein goes towards the minus end, so it would have to be something going towards the minus end of a microtubule, which would be your kinetochore. The one on the kinetochore is going towards the aster, so it's going towards the minus end. Describe 2 positive feedback loops. Okay, so let me pull that slide up so we can look at it together. So chapter 10, which I don't have open. So I kind of laid a heavy hint in my lecture on this, that I might possibly ask this question. And surprisingly, it came up. So here we are, positive feedback loops. Now, some people were telling me about cyclin Ds and CDK4/6, but that's upstream of what we're talking about here. Here we're talking about this step, when cyclin E and CDK2 hyperphosphorylates. What are the positive feedback loops that make this happen fast? One is the fact that once you release E2F, it's going to make more E2F. Another one, there's really 3. The other one is E2F is going to make more cyclin E, and cyclin E is going to hyperphosphorylate more. So there's your positive feedback loop there. The other positive feedback loop has to do with P27. When you have some cyclin E CDK2 activated, it will phosphorylate P27, which then gets degraded. So that's going to activate more cyclin E CDK2, because you're breaking down the inhibitor there. So those are our 2 main positive feedback loops. And that what that tells us is that when you get a little bit of E2F released, you get a really rapid increase in cyclin E CDK2 activity, hyperphosphorylating more, and it has a fast forward effect there. There was also, someone suggested, with the what do you call it, the other transition, the G2 to M transition, there's the phosphorylation and dephosphorylation events, which is another question later. And someone said it's happening here, which is true. And I think I took points off for it. So if I took points off your test for that, let me know, and I'll give you credit for it again, because that does count. Okay, so that's question 3. Question 4. This was one that I gave you in the practice exam. So I like to throw practice exam questions in here just to see if people are paying attention. So a dish of cultured cells were exposed to a drug that specifically inhibited CDK1. At what point in the cell cycle would you expect them to be arrested? And going back to chapter 10, where are you? Right here. Also, this is my favorite figure. So CDK2, no, I'm sorry. Let me get back to that question because I forget which one I said. CDK1. So we've got a drug that's going to inhibit CDK1. Where's it going to inhibit the cycle? CDK1 is CDC2, so that gets activated in the middle of S phase. So that's where you'd be blocked. So someone answered even more correctly than me, if that's, it's just possibly be more correct. I don't know. But anyway, someone said, if you've got a bunch of cells and they're all a different state in the cell cycle, some could be blocked here, right? But other ones would be blocked here. In my mind, I was thinking we're going to start them all at cyclin at G1. Where are they going to get stuck? The first time you use CDK1, which is here. So anyway, that's what we were talking there. Okay, which of the following is not regulated by its degradation? That's cyclin D. right? The other ones are regulated by degradation to keep the cycle going forward. Cyclin D is controlled by transcription through activation by growth factors, and that once the cell gets into S phase, it's no longer responsive to growth factors, and that's what makes it go away. So this is only controlled by its transcription, whereas the other ones are controlled by being broken down. Explain why cyclin B builds up slowly. So again, back to our chapter 10. This is this figure here. Okay, it's building up slowly, but then MPF takes off fast, which is because of this, which is MPF very, very slowly gets activated by CDC25, which is the phosphatase. Once some MPF gets activated, it phosphorylates CDC25 and it becomes more active. And so you have this positive feedback cycle that causes a steep rise of activation. And then, of course, once it's activated, APC gets activated and we break down cyclin B and it goes away. But anyway, this was the key that I wanted to see, which is the inhibiting kinases, the activating kinase, and then CDC25 being stimulated by MPF. Okay. Combination of cyclin CDK hypophosphorylates RB. Hypophosphorylation is D in 4,6. Which of the following is an anti-apoptotic protein, BCL-XL? So we only had two anti-apoptotic BCL2s. One is BCL2, the other is this. And we'll talk more about these today as we did on Monday. How could having one copy of P53 with a point mutation predict a worse outcome than a total loss of one copy? So that again is chapter 10. Well, it's all chapter 10. Why do I keep saying that? Where are we? There it is. So if you've got a mutated but still produced P53, it's going to randomly assort with the protein made from the good gene. And so 15 out of 16 times, you're going to end up with an inhibited P53 complex tetramer. So the key to this was that P53 is a tetramer, and if you've got a mutant as one of your gene, a point mutant as one of your gene, that's going to act as a dominant negative by blocking the activity of the wild type. If you've got a wild type and a null, which means a gene that's not working at all, you're still making normal P53 with the wild type gene, so you're almost normal. Here, this is a little bit less good because somewhere in here you're going to lose that second copy of P53. But with the mutant, it's as if you don't have any P53 at all. Okay, and that can be confusing too, like clinically when you've got a tumor that's got really high P53 and you're expecting it to go into apoptosis, but it doesn't because it's a dominant negative P53. Okay. Choose the proteins that are targets for this should be E3, ubiquitin ligase complex AP3. Shoot, this should be E3. Sorry, guys. I think you know what I meant, though. I hope so. Targets for E3 complex AP3. So that would be securin. Okay, is this more than one, or is this just one? Select two correct answers. Securin and cyclin B. So securin is blocking the activity of separase, which will break down the cohesins. CDC2 is just, I don't know what the hell that. Oh, that's the kinase. That's CDK1, which is just, these are just distractors, just something to throw out there. But cyclin B and securin are being broken down by APC. Get out of here. Where are we? Oh no, that's not APC. Hang on. There. Okay. APC is breaking down securin and is breaking down cyclin B. Okay. when cyclin CDK complex passes through the G2M complex, that would be B and CDK1. That was that one there. CDK1 or CDC2. MYC is a transcription factor that activates the production of cyclin D when it's paired with MEX. Okay, when it's paired with MIS1, it's going to inhibit the transcription of CDK inhibitors. In MAX, it's going to activate cyclin D. And I think MDX is another one which is going to inhibit MYC. And MXD is just something I made up. So that's not a real protein. In the lab, you construct an estrogen receptor with a chimera with P14 to control its movement into the nucleus. Now, the trick here is you have to know P14 is not a CDK inhibitor, right? It's ARF, which has got a totally different function. So what would you expect to see when you expose with estrogen? So a couple of things here. We saw this estrogen trick a couple of times, one with MYC here. And the basic idea is this estrogen chimera, all it's doing is controlling the movement of what it is attached to. So here, without estrogen, it's going to keep that protein in the cytoplasm. When there's estrogen or an estrogen agonist around, it will transfer into the nucleus and it'll have whatever activity it has there in the nucleus. So the question is, if you do that with ARF, what's going to happen? So we're going to go to ARF. What's going to happen is it will be in the cytoplasm until you add estrogen, and then it's going to come into the nucleus. It's going to grab MDM2 and sequester it in the nucleolus. And then your P53 is going to accumulate. So that was the answer there. Okay, order these starting at the end of mitosis and proceeding through the next round of mitosis. So at the end of mitosis, cyclin B and cyclin B has been broken down. And you're going to start out with this guy, cyclin D and force 6. That's going to hypophosphorylate RB and get you up to the restriction point. Cyclin E, CDK2 will get you through that restriction point. And then cyclin E is going to come off CDK2, and cyclin A will jump onto CDK2. So that is what? Number 3. And then cyclin A comes off CDK2 and jumps on CDK1. And then cyclin A comes off CDK1 and cyclin B jumps onto it. And again,
hey, Dr. Slish.
Yes.
Is there any way we can get like a half credit for that one if we had the correct order but started at cyclin B?
Absolutely.
I
actually
did the
did
same
that.
thing.
I did that for a person. Yeah. So why don't you, if that happens, send me an email and I'll give you 2.5 points.
All right. Thank you.
Okay. So Dr.
Yeah.
Slish, because yeah, I did the same thing. I started with cyclin B, CDK1. I guess because I was reading like end of and thinking, okay, we start at the end with cyclin B. So can you just review again the like why you started with the CDK4/6?
Right.
CDKD.
Absolutely.
Yeah. Thank you.
So yeah, I mean, it's a cycle. You can start anywhere. But this is why I started there. I'm like, what happens when you start here? You go from here forward.
Gotcha.
Same thing if we started here and went forward. Okay.
Okay.
Now, and the wording was, I'm going to, wording was starting at the end of mitosis. So it would be at that point. But I understand that you can read that the other way. So yeah, if you did that, then let me know and I will give you half credit for that one.
Okay. Thank you.
Wish. Who the hell proofreads this? Which of the following is not ubiquitinated by Col1? Well, we know cyclin E is, P27 is, E2F is. These all get phosphorylated in S phase. Cyclin D is not controlled by ubiquitination, so that's got to be your right answer. See how I did? 40. I lost 5 points. What? Wait a minute. Which type of microtubule of the mitotic spindle uses Dinaean motor? Let's go to that. I'm embarrassed, frankly. I'm supposed to know this. Okay, so Dinaean motor moves towards the minus. Yep, that one's true. It goes towards the minus. The kinetochore one also goes towards the minus, though. I guess both those.
But it uses both kinesin and Dinaean, so I think that was the trick in that question.
Oh, okay, so it's using Dinaean here, but it's using kinesin here. Yeah, I don't think it's fair. It's not fair. Okay. Okay, guys, that's it for that quiz. Let me show you how you guys did overall. 2. Was this exam 2?
3.
3.
Shoot. All right, get out of here. So there. So most people are up in this area, which is really great. So, oops, oh God, you're not supposed to see each other's grades. Sorry about that. Let's just close this down now. Don't save. Sorry about that. Okay, so that was exam 2. Any questions about that? I just like to say one thing. Patrick, can you make some mistakes next time? It's like, what am I even doing? You don't make any mistakes. It drives me nuts. Give me something. Throw me a bone here. Something I can take points off for. All right. Everyone ready to move on? Okay. Let's go to what we started last week, or Monday. Feels like last week. So we're going to do a little more on apoptosis, and today I'm going to do autophagy, which I didn't do on Monday. Autophagy is probably about 15 minutes. It's complicated, but anyway, we'll move on. So quickly going through what we did on Monday. We went into more detail on caspases and BCL2 proteins and how they end up associating with each other. So they're going to have a pro domain, which is inhibiting the protease activity, and when they get clumped together by attaching to each other, they will cleave each other in 2 different places, giving you a large and a small subunit from each procaspase. And then they rearrange like this, and you've got catalytic subunits and regulatory subunits. And this, like we said, I guess you could call it a heterotetramer, although it may also be a homodimer, because it's like each one comes from the same. I got a phone call here. Let me check real quick. Hello. Sure, but I'm in a Zoom lecture right now. I will check my email when I'm done with that, okay? All right, thank you. Okay, sorry about that. All right, and then we talked about all the different types of caspases. Most of these we're not interested in because they're not involved in apoptosis. These are the ones we are interested in. And we've got 2 different types of initiators, and then these are your executioners. So the initiators are kind of divided into your extrinsic pathways. Would have this death effector domain, and then these have got the other domain, which is called caspase recruitment domain, the card and the dead. So card, and these are going to bind to each other in a homotypic fashion, which means card domains will bind to card domains, dead will bind to dead. So this gives us a clue as to how these initiator caspases are being activated by recruitment, because you've got your TNF receptor or the FAST receptor has got the death domain attached to it as its cytoplasmic domain. And then you've got FADD, which is an adapter protein. It's got the death domain, and it also has the death. Oh God, I keep forgetting this. I'm sorry. Death effector domain. Death effector domain. So then you get death effector domains attaching to each other, and even within caspase 8, and now they're close enough that they will cleave each other. And this death effector domain is part of that pro domain that gets cut off. So they cleave each other and they form that activated initiator caspase. Similar thing over here with our apoptosome. APAF1 has got a card domain, which is going to be hidden when there's no cytochrome C around. This will fold up and hide that card domain. When cytochrome C is here, it opens up, it then binds ATP, and it stretches out, and then your card domains associate to form that apoptosome. And then again, while you've got your caspase 9s close to each other, these are going to fold over on top. Then they're going to cleave each other and activate. Let me take a quick look at that. Oops. So here's what that apoptosome looks like when it's all put together. And the purple part in the middle is all the activated caspase 9 that gets accumulated together and activates itself. Okay, so that's our caspases. And then once you get your initiator activated, these then become a target for the initiators. You're just going to chop that pro domain off and then cut it here, and then they reorganize to form the active executioner caspases. Now, targets of those executioners. PARP, which is a nuclear protein involved in looking for DNA repair, DNA damage, cytoskeletal proteins, and this is what's going to cause that cell to collapse like it does during apoptosis. Gelsolin gets activated, and it starts to break down microfilaments. Actin itself, part of the microfilaments, is getting broken down. Vimentin, part of the focal adhesion structure. So that's going to cause it to let go of the cells around it, or the extracellular matrix around it as well. So all that's being broken down, and that's rounding the cell up now because it doesn't have cytoskeleton anymore. And here, note, if you were here Monday, I had a typo here. I had ICADs here. This should be, oops, I'm sorry. This should be IAPs. So I've corrected this. I will upload this PowerPoint after I'm done today, and it will be corrected there. But this was a typo last time. Inhibitors of caspase proteins. IAPs. So anyway, these are proteins that we hadn't talked about previously, but these inhibit our caspases, both the executioner and the initiators. So again, with cells, it's always a balance between cell division and apoptosis. And you've got proteins out there associating, associated with both processes. And when you activate cell division, you inhibit the apoptosis. When you activate apoptosis, you inhibit cell division. So those proteins will be inhibited as well when we get into this mechanism. And then signaling molecules. Like I've said before, we look at signal transduction. When you activate cell division, you're also inhibiting apoptosis through all these mechanisms. So your caspases then are going to break down AKT. So you can no longer block BAD. It's going to inhibit MAP kinase, and MAP kinase also, MAP kinase are also going to be activating, blocking apoptosis. And you're going to break down BCL2 and your ICAD. So another thing we didn't talk about in the last chapter, you've got these proteins in the nucleus as well that are inhibiting the DNase that will break down the DNA. So here's our IAP story here, where you've always got IAPs there inhibiting your pro-caspases and your caspases. And you've got this protein SMAC. And again, two different names, SMAC and Diablo. So SMAC Diablo is in the mitochondrion being blocked. Its exit is being blocked by BCL2. At the same time, it's blocking cytochrome C. And then when you get activation of the intrinsic pathway, we inhibit BCL2, we release SMAC Diablo, and SMAC is going to inhibit your IAPs, which is allowing the caspases to be active, and cytochrome C is making your apoptosome work. Okay, so the only thing we've added here to what we said before is the IAP SMAC Diablo story. BCL2 proteins, how are these things working? I'm glad you asked. So we've got three different groups. Group 1 is the anti-apoptotic BCL2s. Structurally very similar are the multi-domain pro-apoptotics. And these have got 4 different domains of homology between them. And the original BCL2, so we call this BCL2 homology 4, 3, 1, and 2. This guy here is probably the most important one, BH3, because we're going to get that homotypic dimerization between these domains here. That's going to be part of the mechanism that activates things. So these guys here are the ones that are going to, see, they've got this transmembrane domain here. These are the ones that are going to go into the mitochondrial membrane and allow cytochrome C to release. And these are going to be activating these guys. So these are the pro-apoptotics that get activated by different mechanisms, as we said before, PUMA, NOXA, BAD, and BID. So we'll look at BID here. How does BID get activated, and how does it cause apoptosis to start? So BID is self-inhibited here by a domain that blocks its activity. And when we get the death signal here, in this case, I'm using tumor necrosis factor alpha. So TNF alpha then activates the TNF alpha receptor, which then attracts FADD and caspase 8. So that's what we saw earlier with the death domains, the death excitatory domain, or death, I always forget what the E stands for. The DD domains, DED domains are all clumping together to give us caspase 8 activation. 8 then cleaves off that pro-domain from BID and becomes TBID. TBID then activates BAX and BAC and inhibits BCL2. I think the exact mechanism of how it does this isn't quite understood yet. So we're not going to worry about that. But when we do inhibit BCL2 and activate BAX and BID, they're going to insert into the membrane and form a channel. Cytochrome C is coming out, SMAC's coming out, and then we're activating apoptosis. So the key thing here to keep in mind is these homology domains that are going to help them to dimerize. And that's what causes activation. Okay, and I just talked about this. All right, so find me and eat me signals then. Once the cell has gone through apoptosis, those apoptotic bodies are asking phagocytes to come and get them. Now, phagocytes, we've got dedicated phagocytes, which are macrophages and dendritic cells, and induced phagocytes, which are endothelial cells, epithelial cells, and fibroblasts. So either of those can be stimulated here. And it's important to get rid of all this apoptotic bodies fast because they cause inflammation, and inflammation is bad. Okay. So find me signals are chemoattractants. So we've got lysophosphatidic acid, which is basically, lysophosphatidic choline. So here's your choline phosphate, here's your glycerol backbone here, and all we did was take that one fatty acid chain off, and we get LPC. So LPC then is a chemoattractant, This is sphingosine phosphate. So sphingosine is another membrane lipid. Gets phosphorylated and released. And then this is fractalkine, I think, fractalkine, which is a cytokine, another attractant, and nucleotides can get released. So basically, these things are then going to activate receptors on the surface of the phagocyte, cause amoeboid movement and movement towards those apoptotic bodies. And then once they get there, the eat me signal. Main eat me signal is our phosphatidylserine. And again, we talked about this back in chapter 2. Your caspase 3 is going to cleave both the flipase and the scramblase. When it cleaves flipase, it turns it off. And flipase is putting phosphatidylserine on the inside of the membrane constantly. So that gets shut off. And then you activate scramblase, which just scrambles them so it's random. So now the half the PS is on the outside, and that phosphatidylserine then becomes a receptor for proteins on the phagocyte to grab that apoptotic cell. And here's another look at that. So here's your phosphatidylserine sticking out, and receptors on the cell are going to grab it, and then it's going to like surround it, bring it in, and fuse it with the lysosome to digest that apoptotic body. Okay, so that is the eat me signal. It's basically phosphatidylserine, as we talked about in chapter 2. Okay, that's it for what I did on Monday, 11:30, and I feel like I need to take a deep breath here. I've been going pretty fast. All right, so is everyone good with that? Everyone cool? All right, so let's move on and talk about autophagy. What is autophagy? So whenever I think about autophagy in cells, I always think about like concentration camp people, how they don't get enough or anyone's stranded where there's no food, right? You don't get enough nutrients, so cells start, your whole body shrinks down, and the muscle proteins get eaten up. You're basically eating yourself to try and stay alive. And you're not losing cells, at least in terms of muscle cells. You're not losing cells. You're losing proteins inside those cells as they're, breaking down proteins for energy to try and stay alive. Same thing here with the cell. I mean, it's the same process here. But what's going on with the cell is, two things. One is housekeeping. And in terms of housekeeping, what I mean is you'll have chunks of broken down organelles that don't work anymore, or you could have just proteins accumulated together that are non-functional. All this stuff has to be cleared out to keep the cell efficient. So that's the housekeeping idea. And then survival, which is more what I'm going to be talking about today, is when nutrient concentrations are really, really low and you need to start breaking down what you have in order to survive. So keeping that in mind, what's going to activate this autophagy in terms of survival is a decrease in the energy available in the cell. So when there isn't much, there aren't any, sugars and things to break down, your ATP levels drop, which means your AMP levels are going to come up. So if you've got, some ADP in the cell, you've got your energy stores in the cell. You've had biochemistry, I believe, but your energy stores of quick, high energy phosphates are ATP is what's being used. It gets broken down into ADP. That ADP can then be broken down, give you AMP, and recharge an ADP to an ATP, right? So you can transfer that phosphate over so you can keep your ATP levels high. But when you do that, AMP increases in the cell. And also you've got your creatine phosphate, which is another buffer for high phosphate, high energy phosphates. But bottom line being, when you use up all your ATP, you start converting ADP into AMP and ATP. So now you end up with a lot of AMP around. And AMP is going to activate this kinase, which is called AMP activated kinase. And so this kinase is then measuring energy level in the cell. So keep that in mind. We'll come back to it. So we've got as always, we've got two different pathways. One is things are fine and we're growing. And the other is we're running out of energy. We need to conserve. We need to start breaking things down. And the happy high energy pathway is our mTOR pathway. So let me back up to chapter 9. Okay, this keeps coming back. Here is mTOR Raptor over here. So AKT is going to inhibit TSC, which activates mTOR, which is activating protein synthesis and cell growth. So this pathway is all about cell growth. And so when the cell is starving, we don't want pro-growth signals going on. So AMP will phosphorylate mTOR. It'll come off the mTORC complex and become inhibited. So we'll stop that pro-growth pathway. And we're also going to activate the autophagy pathway. And this is the key step. One of the key steps here in autophagy is activating this ULK1 complex. You want to think of ULK1 as the trigger for autophagy because this is going to start it all off. So when everything is good and we're in a pro-growth mode, pro-growth mindset, as we say in business, it's a pro-growth mindset. The mTOR is inhibiting this autophagy pathway by phosphorylating ULK. Then when we go back into survival mode, AMPK is going to inhibit this and activate this. Now this ULK1, the interesting thing about this is just how you've got every different kind of signal transduction going on here in this one pathway, which is kind of weird. So this is a kinase, and this kinase then is going to activate Beclin-1 complex. So there are a lot of proteins in this Beclin-1 complex, which are being activated by phosphorylation here. One of them is VPS34. VPS34 is like PI3 kinase. Let's go back to chapter 9. PI3 kinase, right, is going to phosphorylate PIP2 on that 3 position of the inositol and give you PIP3. Well, this VSP1 is going to do the same thing, but just on PI. So let me blow this up so we can see it better. You know what? I am on my own computer and I can do whatever I want. So I'm going to do this. Let's look at this. So here's PI3, PIP3 or whatever, right? Phospholipid. Here's your glycerol backbone, 2 phospholipids, phosphate to inositol. Now this inositol ring, 6-member carbon ring, is numbered 1, 2, 3, 4, 5, 6. Typically, PI doesn't have any phosphates on the inositol. PIP is usually phosphorylated on the 4. PIP2 is phosphorylated in 4, 5. And then when PI3 kinase comes in, you've got triple phosphorylated PI. What we're doing here is we're not putting any phosphates over here. We're only putting it here. And that's going to make it different from, usually we say PIP, right? PIP is phosphorylated on the 4 position. So this we're going to call PI3P because to distinguish it from the normal PIP that's in membranes. So that's what's going on here. Get this guy out of our way. Okay, so kinase activated by kinase activating a PI3 kinase, but not a protein kinase, a phospholipid kinase, and that's going to initiate our autophagy. Now, how does it do that? You're going to be so excited when you hear. So we activate ULK, activate Becklin and VSP3. You're making PI3 phosphate. Now that's going to start this isolation membrane growing off of the rough ER. So this is a rough ER mediated kind of a thing. So this isolation membrane, you can imagine just the membrane coming out, and you're phosphorylating PI in the membrane to PIP2. That then becomes a binding site, just like PIP3 was, for WIPI. So this is going to be an adapter protein that's going to bind to the isolation membrane based on that phosphorylation of PI by VSP34. WIPI then is going to bring in these two enzymes. These are both E3 ubiquitin ligases, ATG16L and ATG3. And so ATG16L, and I don't know if the mechanism is known all that well, but this is ubiquitin ligase, but somehow it's going to activate this ubiquitin ligase. So ATG is a ubiquitin ligase that's then going to put this ATG8 on phosphatidylethanolamine. Now tell me that isn't freaky. That's kind of weird. So this ATG8 is ubiquitin-like. So this is an E3 ubiquitin ligase, but it's not using ubiquitin itself, it's using ATG. A lot of sequence homology between ubiquitin and ATG8. And it's attaching it not to a protein, but to a phospholipid, phosphatidylethanolamin. So this is going to mark this as the isolation membrane, because now this will be the target for, or the receptor for things to be accumulate into this isolation membrane to turn it into the autophagiosome, which is what we're getting to next. Okay, so let me just do this again. Activation, low ATP activates this, which turns on this kinase, which turns on this phospholipid kinase, which gives us PI3 inositol. And then that activates these ubiquitin ligases, which takes this ubiquitin-like protein and attaches it to phosphatidylethanolamine. Okay, so that's now in our isolation membrane. Okay, so this is our isolation membrane here, and it's got ATG attached to phosphatidylethanolamine. So it's kind of like it's got receptors all over it, waiting for things to bind to it. These guys here, NIX, P62, NBR1, these are going to be adapter proteins. They'll bind to ubiquitinated things on this side and to ATG8 on the other side. So these adapters then are going to bring things like damaged mitochondria and big clumps of misfolded proteins into this autophagiosome. Okay, so once that starts to happen, you start accumulating things inside this isolation membrane, and it will eventually surround the entire clump of whatever it is you're getting rid of here and form the autophagiosome. Now that autophagiosome then will fuse with the lysosome. We'll get release of the acid hydrolases, activation of the V-type hydrogen ATPase pump, acidifying the inside of the membrane, and then breaking everything down. And then releasing those monomers, which can be used for energy or building other things. Okay. Whew. Autophagy. So quick review of autophagy. So this is kind of the too long didn't read version. Activation of AMPK by AMP leads to activation of VSP34. And that's going to give us our PIP3. Remember this guy? Okay, PI3P. PI3P then brings in the ubiquitinating enzymes, through WPI or WIPI. And those are ATG6L and this. Okay. ATG3 then attaches the ubiquitin-like molecule ATG8 to phosphatidyl and ethanolamine. And then this becomes a receptor for material to be engulfed. And then things come in and be engulfed. Autophagisome forms, and the engulfed material fuses with the lysosome. Ooh, dog. That's a lot. So let's pause for a second here and talk. Oh, Michaela, nice picture. So how do we feel about that? Is that okay? Is that good?
It's a lot of information, but it's
a
good.
lot.
I think that was really good.
Okay, so read over that again next Monday. I will review the autophagy part again. And now, if no one has a complaint or a compliment or anything like that, I will move on and start talking about cancer. Are we good? Good to move on to cancer? Everybody loves cancer. Let's do cancer.
Sounds good.
I'm really glad we split everything up.
Yeah. Can you imagine that on top of the other material for exam 3? And I was going to do, I've got, how many slides do I have for cancer? I've got 73 slides for my cancer lectures, and that was supposed to be in that last lecture material. Definitely too much. Okay, so just to let you know what I'm thinking, because as far as I can tell, you're not reading my mind here. The next exam will be this chapter 11 that we just covered most of here, and then this cancer stuff that I'm adding in now, which is going to be probably 2 lectures. And then I'm going to go into signal transduction used in early embryo development, because I think that's really fascinating, the way changes in gene transcription causes differentiation and then cell movements and all the stuff. That's really cool stuff. So we'll do that. See how far we get. That probably will be all we do in this class. If we've got more time, I've got more things I can talk about. So anyway, that's the plan going forward. So let's talk.
So does that mean that the rest of the material that we cover will be all on one exam as well?
Yeah, right. Well, whatever we cover from the last one coming forward will be on the last exam. We do have quite a few lectures left. I don't want to overburden you with material. Maybe we can do some, if I can get some feedback from you guys, we can do some more practice questions that we can talk about in class. But what I don't want to do is give you a practice exam and then in the next class just answer all the questions for you. I want to get some answers from the crowd as well. So we'll see if we can do that in the next class or the one after. Oh, and again, speaking about the next class is during the eclipse. And so, they're telling us not to drive anywhere. Dr. Lounsbury told me to do a lecture and just post it so it wouldn't be synchronous. I'm going to go ahead and do a synchronous lecture anyway. You don't have to come to it. I always post them anyway. But Monday I'll do an online lecture. I'll do an online lecture. Although you can come or you cannot come. It's up to you. So I guess that's all I wanted to say. All right. Any questions, or should we dive into cancer for about a half hour? Let's do a little bit of cancer. By the way, I hope you don't think I'm being flippant about cancer. I mean, my mom died of cancer, so I'm well aware it's not a fun thing to joke about. But here we go, chapter 11. Okay, so I'm calling this chapter 2.10.2. Chapter 10 in the book that I assigned talks about cancer, but it kind of goes over briefly and it hits certain points in detail without really explaining the connections between the two. So I'm kind of freewheeling it here. I'm going off on my own. There is a book that I ordered for the library there, the Dana Library, called Biology of Cancer by Weinberg. That's where I'm getting a lot of this information from. So if you want to read it in the textbook, you could do it there. Although keep in mind, anything I talk about here, you can do a Google search. Another thing I like to do is go to ChatGPT, so I'm like, how does this mechanism work? You get to Wikipedia, doesn't really explain it. Go to ChatGPT, ask the question, you get a pretty good answer. Sometimes it's not right, but it gets you closer anyway. So if you're if you don't want to use the textbook, the textbook I assigned is not really good for this. You don't want to use that Weinberg textbook. Just do some Google searches and ChatGPT. There's nothing new here. This is all settled science. Okay, so cancer biology. We're going to talk about changes in cells as they go towards cancer, and then mechanisms that make that happen. So first, kind of broad general categories of different types of cancer cells. And this is kind of Bio 101 again. You've got benign cells, which are not considered cancerous, but they are wrong cells, right? These are cells that are dividing too much and they're forming a tumor. And the key thing to benign cells here is they are all kind of contained in one area. They're not moving around, they're just contained in one area. And if you're lucky enough to have it diagnosed when it's like that, you can just cut it out. Some tumors will never get bigger. I mean, how many people have dogs and you pet them and you find a big lump? It's just a fatty tumor, that a lipoma, and it's never going to go anywhere. They're just not dangerous cancers. So those are your benigns. And then you've got malignant. Now, malignant is when they cross this basal lamina. So this is something I'm going to be harping on in this chapter, which is the protein barrier between epithelial cells and the stromal cells that are underneath the epithelium. Big, thick protein barrier here. It's either called the basement membrane or the basal lamina. When cells cross that, that's when they're malignant. And that's when they can start moving around and destroying tissue. And that's when they're dangerous, right? Because, I mean, if you've got a lump on your breast and it's just a lump on your breast, so what? It doesn't matter. But once they start moving and then they start to go metastatic, that's when you start disrupting important tissues. And that's when they get dangerous. So malignant cells then are crossing that basement membrane and starting to move. And then they can move their way all the way into either a blood vessel or a lymph vessel. And that's when they become metastatic, which means they're moving and setting up new tumors elsewhere. And like I say, you've got a lump on your breast, whether you're a male or female, doesn't really matter. But when it spreads to your lungs, that's when you're in trouble, because it's going to disrupt the function of the lungs. And we'll talk later about why they tend to go to the liver, the lungs, or whatever. But basically, metastasis is when they're starting to travel through the lymph or blood and set up new tumors. Okay. All right, tumor progression. So I am not a cytologist. I'm more of a molecular guy. So if you guys know more histology than me, please don't be afraid to speak up. But to look at the difference here between normal cells and as they're getting more and more dangerous and eventually cancerous, we start here with hyperplasia. So hyperplastic cells are basically just normal cells that are dividing too much. So they look just like normal cells, but they're disordered because they're no longer in the position they're supposed to be, and there's too many of them. So this is an early hyperplasia in the breast tissue. So here's normal. You've got a basement membrane, which you really can't see, but it goes right around here. And within that basement membrane, you've got two types of epithelial cells. You've got your glandular cells, which are can't remember the name of them. When they're cancerous, it's adenoma. So I'm sorry, I can't remember the names of them. But then your regular structural epithelial cells. So you've got a mix of both of them in your milk duct. And those glandular cells are making milk proteins and things. And the other ones are just structural epithelial cells. And you've got a big lumen here in the center that can be filled up with milk and stuff. So here is your hyperplasia, where you've got these cells that are growing into the center of that lumen and blocking the lumen. So you no longer have a nice big open lumen. So basically too many cells, but they're all pretty normal. The next step then is metaplastic. And this is when you're crossing barriers, but not that basal lamina barrier, right? So you're just crossing from one place to another. So this is what's called Barrett's esophagus, when you've got cells that are stomach cells that are crossing the cardiac sphincter and going up into the esophagus. And this can happen when you've got chronic GERD, right? Gastroesophageal reflux disease. You've got acid constantly coming up this way, and that's causing these stomach cells to come up into the esophagus. Here's an actual picture of what that looks like. These pink cells here are not supposed to be, or the red cells are not supposed to be here. The pink is normal. The red is cells that have moved through this gastroesophageal junction and up into the esophagus. So basically, metaplastic cells are in the wrong place. Another place you can see this is like in the cervix, when cervical cells are going up into the uterus or down into the vagina. So crossing a barrier, but not this barrier, which is your basal lamina. Dysplastic. Now, dysplastic is when the cells themselves are not looking normal anymore. So your basal membrane is still intact here, but they're pushing down in. So that's your benign tumor, right? Because it's you've got too many cells to be normal, which would be a thin layer of epithelium up here, and they're pushing down into the stromal layer, but they haven't broken the basement membrane, and they're staying in the epithelial side. So your cells are different. They're bigger. They're rounded up. They can have 2 nuclei or large nuclei, and the nuclei problem is telling you that there's some chromosomal changes now, and chromosomal changes are really dangerous. That's what makes your cells become more and more dangerous. So what was I going to say? Oh, remember our P53 is supposed to make cells die when you've got chromosomal problems. Here, that's not happening. So the cells just continue to divide when they've got serious sickness to them. And then papilloma is similar in that you're not crossing the basal membrane, but you're going up this way and forming these papillomas that stick out. I think like a wart is a papilloma, so it's got a blood supply and it's growing bigger, but it's not going to go back and then into the body. So at this point, you're is still benign. The next step then is neoplastic. So neoplastic is when the cells are just plain, like all the cells in the area are just plain not normal. But they haven't crossed the basement membrane, so it's in situ. So carcinoma in situ. So they're basically cancer cells, but they haven't crossed the basement membrane, so they're still in their normal place. So dysplasia, when the entire epithelium is dysplastic and no normal epithelial cells are present, then the process has gone beyond dysplasia and is now neoplasia. If the basement membrane is still intact, as shown here, then the process is called carcinoma in situ, because the carcinoma is confined to epithelium. Neoplastic epithelium is termed carcinoma. So this is a carcinoma, even though it has not crossed the basement membrane yet. Okay, and then next step then is neoplastic invasion. So these cells have gone through this basement membrane and are now invading the stromal layer of tissue and starting to move around. And again, this is when they get dangerous because now they're on the move, and you know, skin is skin. Who gives a darn if you've got a bump or a lump on your skin? But once it starts going into the body, that's when it starts disrupting important things. And we'll talk about this later in this chapter, but the way cells are going to get into this body, it's not easy, right? Cells are not just floating around in fluid here. Cells are attached to each other by cell-to-cell contacts, by cell to extracellular matrix contacts, and you've got extracellular matrix around the cells. So you can't just, like, a cell can't just walk through all of that. You need to start putting out proteases and breaking down all that extracellular matrix to allow these cells to move. And then the next step is metastasis. So you've got an awful lot of barriers that these cells have to be good at crossing in order to become metastatic. So first of all, they have to break out of their the epithelium and go into the stromal layer. Once they're in the stromal layer, they're crawling through extracellular matrix until they get to a vessel of some sort, and then they have to crawl into that vessel. And once they're in the vessel, they're going to float around free. And as we talked about previously, and we'll talk about again, cells shouldn't be floating around free, right? Only blood cells should be allowed to float around free. These guys should be connected to extracellular matrix. So they've developed enough mutations that they can get away with it. And we'll talk about that later. But self-sufficient growth signals allow them to survive in solution, which they shouldn't be able to. But anyway, they're floating around in the blood, and then eventually they're going to come out of a vessel, blood or lymph, come out of that vessel and set up new tumors. And this again is when that cancer is going to kill you. And also, when the cancer is still kind of in one place, the therapies are directed to that spot. So, you know, your concentrated x-rays on that spot or surgery to pull it out. Once it goes metastatic, now you're talking chemotherapy, because you can't just irradiate the whole body, not easily anyway. All right, so we've gone into blood vessels and lymph vessels. So where are we going to metastasize to? Some types of tumors are attracted to certain areas. Like one example I know of is prostate cells, when they metastasize, tend to go to bone tissue for some reason. I don't understand why. I don't know if it is known, but I mean, that's one little factoid that I know. Some cells will go to certain areas, but in general, where cells are going to go is the next capillary bed. So if a cell like a melanocyte gets into the blood supply, the next capillary bed it's going to see will be the lungs. So, you know, the blood will be collected from the skin, go to the right side of the heart, from the right side of the heart out to the lungs. And on that whole trip, the vessels get bigger and bigger and bigger, right? You get to the lungs, they get smaller and smaller and smaller. And now you're in a capillary bed and the cells can escape and then set up tumors in the lungs. So that's one reason that you often get lung cancer metastasis. Also, let's see, do I talk about, no. Also, you know, if you've got cancer cells metastasizing from the small intestine, large intestine, remember that hepatic portal vein, right? Everything you eat goes through, gets absorbed, goes through the hepatic portal vein to your liver, where it then goes through a capillary bed and gets filtered, right? Your liver's filtering out toxins and stuff. So if you've got cancer cells that are starting in your intestines, they go into the blood supply, they go to the hepatic portal vein, they end up in the liver, and you get metastasis to the liver. So the liver is another really important organ that can get metastasized to. So liver and lungs are targets. On the other hand, for your lymph, if you know, lymph is soaking up all the extracellular fluid around, and if your metastatic cells get into that, they make their way up to the lymph nodes. And now here at the lymph nodes, again, they're going to get slowed down and stopped. And they'll fill up this, and then they'll set up a tumor here and fill up the lymph node with cells. And that's why often with someone's got a lump on the breast, first thing they want to do is look at those lymph nodes. Is it metastatic? Is it starting to move? Because breast cancer will often metastasize through the lymph, and then the lymph nodes are the first thing that end up getting attacked. So that's it. I'm going to review in a second, but that's about it in terms of metastasis. You've got a lot of protein breakdown and movement going on, and you're most likely going to end up in the lungs or the liver or in the lymph nodes. Unless, like I say, there are other things going on that attract those cells to other tissues where they will set up. I mean, you know, cells will metastasize to the brain as well. So there are other metastatic sites, but these are the most common for those reasons. Okay, cancer cells. So this is just kind of review of what I said. You've got normal cells, and you've got a cell gets a mutation. It starts to divide. It becomes a hyperplasia, too many cells. Dysplasia, weird cells and too many of them. And then they become cancerous, cancer in situ because it hasn't crossed the BM. And then invasive cancer crosses the BM and then possibly metastasizes. Okay. Any questions about that? I think if it's okay by use guys, that's Pennsylvania speak. I was in Pennsylvania this weekend, so use guys. If it's okay with use guys, I think I want to stop there. What we're going to do, where we're going next, just to steal my own thunder, is we're going to talk about how cancer cells are different than normal cells, what changes happen in them. And we'll talk about all of these different processes that happen that cause them to become metastatic and such. Okay, so we'll do that on Monday. And I'm back. Okay. So I think that's about all I want to do today. Do you have any questions from the crowd out there? Any comments?
It's not over material, and I know we only have 1 month left, but do you think that for the quizzes, you could potentially start opening them a little earlier instead of just on Fridays? I don't know if you write them like when you're finished writing them, but
I write them on Thursday, but I could write them on Tuesday or Wednesday. I mean, if you guys want them, what do you want? Like what do you think would be good? When should it open? It's like this is my first time through this class and doing this online with you guys, so I'm looking for feedback here.
Yeah. I mean, I would understand that since we finish usually the material of the week on Wednesday, if you needed to take a day and then just did it on a Thursday, even that extra day would be really appreciative. I don't know how other people feel. I just know that I work on the weekend, so it's usually trying to cram the quiz in.
That would be good, because then after I finish my Wednesday lecture, I'll know what we've covered up to now, and then I write the quiz, and then it'll be open Thursday morning. Okay?
I agree with opening a day early as well. That gives me time because I work on the weekends as well, so that works for me too.
Cool. That sounds great. So I'll start that today. So tomorrow the quiz will open, which means I better write a damn quiz today, shouldn't I? Okay.
I appreciate that. Thank you.
Sure. Not a problem. Not a problem. Any other questions, comments?
Mr. Donald, I would like to speak to you when everybody else gets off the Zoom.
Wait, I'm sorry. Could you say it again?
I would like to speak to you when the other classmates get off the Zoom.
Okay. So you want to Zoom together, just me and you?
Yeah.
Okay. This week's busy for me because I've got advisement students coming by. So I'll put you into my Google Sheet for advisement. Just choose a time and I'll Zoom with you at that time, okay?
Okay, that'll work.
Yeah, like normally I can say, yeah, anytime Friday because I'm not busy, but a lot of my time Friday's been taken up by advisement things. So I'll attach you to that,
Okay,
site.
well, I sent you over an email this morning. I don't know if you got it because I did lose power here for a second in Nashville, but I did shoot you over 2 emails this morning.
Oh, there it is. I see it.
Okay.
Yeah. Oh yeah, that's a good question. She says one of her questions wasn't graded. Yeah, that happened. I don't know. Something's weird about Brightspace. Sometimes I'm not given the question, I don't grade it, and then you get a 0. If you get a 0 on a written question, that means I didn't see it. Because if you've written anything, you're going to get 3. That's my grading scale. It's like, I want to give you 3 points just to encourage you to keep trying. So even if it's ridiculous, it's like, at least you're out there trying. So if you get a 0 on a written question, that means I didn't see it, and it automatically was given a 0. Okay, any other questions?
And yeah, and then that was it. That was the only question about my test. And then my exam 3rd grade is not showing up at all. I don't know if it's because I had a glitch in my bright spot over the past week. I had technical issues over the past week, but it's not.
When did you do your exam? Did you do it yesterday afternoon, evening?
No, I did it on the 1st.
Yeah.
What today is?
Well, today's the 3rd, so
Yeah, I did it on the 1st.
Okay. Yeah, I'll look into that.
Okay, thank you.
Okay. Anything else? Okay, guys. Let's call it a day, and I will see you again on Monday. I'll see you before I see the eclipse and lose my eyesight. All right.
Wear your
Bye-bye.
glasses, Dr. Flesh.
Where are my glasses? Okay.
Thank
Have a good
you.
one.